The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and thank you for joining us today. You've logged into the Nelson Labs webinar, ISO 10993-1 panel discussion, preparing for updates in the new FDIS. Now, if you miss one of our webinars or would simply like to refer to one, you can always find them on the Nelson Labs website, on-demand webinars, on the on-demand webinars page listed under education. You can receive notifications for upcoming live and on-demand webinars by liking Nelson Labs on Facebook or following us on LinkedIn. We also invite you to register for the free Sotera Health Academy at soterahealth.com/academy. There you'll find curated content for industry thought leaders, from introductory sterilization and lab testing topics to advanced in-depth learnings. Our expert advisors filled this academy with cutting-edge educational content and resources to help you mitigate risk, go to market faster, and achieve excellence in your field. Nelson Labs is a leading global provider of laboratory testing and expert advisory services for medtech and pharmaceutical companies. The company performs over 900 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and protective barrier industries. The experts at Nelson Labs know every test matters and requires solutions to complex problems to improve our patients' outcomes and minimize client risk. Now, let's get started. Today, we're joined by Thor Rollins, Chris Parker, and Nicholas Cristiano. Thor is a certified microbiologist and specializes in the selection and conduct of in vitro and in vivo biocompatibility tests, and he actively speaks on biocompatibility-related topics through Nelson Labs external seminars, webinars, and trade shows. Chris is a biocompatibility expert with almost 20 years of experience working closely with medical devices and combination product manufacturers to develop their biological planning and evaluation programs with global submission support. Nicholas serves as the global marketing segment leader for the biologic safety here at Nelson Labs with over 12 years of experience in the medical device biocompatibility field. We do encourage your questions, so please submit them at any time in the question box within this app. Thor, Chris, and Nicholas will try to answer as many as possible. Now, to begin today's panel, I turn it over to the group.
Go ahead, Nick.
So, as Brennan mentioned, my name is Nick. I'll go ahead and get us started here today. Thank you, everybody, for taking the time to join us. As mentioned, we hope to get through as many questions as possible to help everybody understand these dramatic changes that we're faced with currently. So just a quick agenda for today. Chris, Thor, and myself will go through a couple slides. We want to keep it brief with high-level topics and high-level areas that we believe at this standard are most worthy for us to spend our time talking about. So as we set the stage with a few of these topics, please continue to submit your questions. We'll come back to the chat box and we'll go through as many as we possibly can, as mentioned. So to start off, why we're here today, talk a little bit about the timeline to publication for these Part 1 changes. As we're all aware, we currently have been working under the supervision, if you will, of Part 1 2018 revision. And so this new product register, or basically a discussion to change this revision, update the revision, came to about August of 2021. And so in August 2021, the discussion was started to make changes to the revision and to move forward from there. A working draft was submitted about a year later in September of 2022, which is when the first time that the committee basically sees this draft. So now fast forward about a year, as mentioned, to September of 2022. The ISO committee experts then see that draft once it is balloted in July of 2023. And from there, we have about a 1.5-year mark, or about 2-year mark now, until we have reached the publication stage of the standard. And so we're going to talk a little bit today as to what the U.S. position has been on this standard and why. And why I'm talking about some of these random dates along the timeline is to help set the stage of understanding why the U.S. has felt like we moved pretty quickly through this revision. And so again, setting the stage for some of these further discussions. The ISO committee experts basically saw this new revision back in July of 2023. About 1.5 years later, we get to Paris in October of 2024, where the standard had been voted to move to FDIS stage. And so for those unaware, the FDIS stage is basically a point when the standard is completely finalized for the exception of editorial changes. So technical changes at that point would be considered locked in, if you will. And so now from the end of 2024 until now is when we reach the publication stage. So again, I know I threw a lot of dates at you, but really understanding that we move pretty quickly from previous revision to committee for seeing the standard, and then a 1.5-year stage of technical discussion of those changes within the standard. So again, a pretty small amount of time for a very large update to what we're going to discuss today. Within those updates include, again, not an exhaustive list, but some things we find to be most valuable to spend our time on today. Adjusted effects to be evaluated, including genotoxicity requirements, MMP being removed from the standard. So I would definitely consider this a win for most of us here, for the exception of maybe novel materials, which we'll talk about lightly if we have time, and then new guidance on cumulative contact duration. So really putting emphasis on understanding and properly identifying what contact durations are for our products moving forward. It also recategorization of repeat use and absorbable devices. So we'll talk a little bit about that today as well. And another large area in life cycle management or end of life evaluation. So determining not only the useful life of our products, but then also what are the biocompatibility risks along the way within that useful life.
All right, thanks. And to touch off on a couple more of the details, there's an awful lot that went into the document to give a little bit more of a framework of some of these risk evaluation documents when it comes to the biological evaluation plan, the evaluation report, biological risk assessments, which draws in a little bit down below with a point about biological equivalence, and then also the toxicological risk assessments. It gives a little bit of a framework of some of those documents so that hopefully there's a little more consistency between writers and who is doing these documents. So there's a framework that sets up across ubiquitously. But then further there's an increased requirement on who can be an author on some of these documents. We'd heard from some notified bodies and other regulators that some of the BEPs and such that they were receiving weren't necessarily telling the full story that the regulators were hoping to receive. And so there's a requirement for both formal education, training, continuing education, and so on. And so working with some of the folks like our experts here at Nelson for getting those documents written will be even more important from those qualification backgrounds. You're going to have to submit CVs of the authors. There's an ever increasing connection between the 14971 and the risk evaluation and how we look at risk estimation, risk analysis, and so on. Then finally, how much risk is too much. And this is one of those contention points that a number of folks had concerns about. And there's actually being an expedited review of trying to get a formal document put together on that. Normally those take upwards of 3 months to write, but we're hoping to get one written in 8 weeks to help supplement the current standard that's going to be published over the next 6 to 7 weeks. There's a lot more about foreseeable misuse. And again, this is another point that there's a little bit of concern in regards to, because how far do you need to go down the rabbit hole of misuse? You need to consider every single possible off-label use, or what is the reasonable level. But we do need to consider your device being used in other indications for longer durations and under other clinical environments than it might otherwise have been intended for, and just labeling against it might not be sufficient. There is a great piece in here is that unlike with the MDRs, you don't need to go reevaluate your devices. The hope in the design is that you would look at the life cycle and sort of identify some of the gaps, but it's not to go back to do a full reevaluation or retest or anything like that. It's to use the information to help support the future life cycle and iterations of your device. Biological equivalence is looking at how to draw a comparison between a predicate device or a comparative device and your own to try to support your submission and the risk evaluation, and maybe not having to test to as many endpoints and be able to risk assess between them. And then finally is this recognition in the state of the art. And this is one of the major points that we'd heard from people about in some of the pre-discussions, and we've heard directly from industry, is how quickly should we expect once the new standard publishes in the next about month and a half or two, is now where am I going? How quickly will FDA or notified bodies or China or Japan or so on be to start expecting these changes to be implemented? And we know that in Europe and other regions, they will consider it as state of the art, meaning that they will start to require the implementation of the new -1 standard almost immediately, which means if you have ongoing programs and you're going to Europe, and first in the next short duration, that you're going to want to start this implementation and bringing in these elements. But we don't know from the FDA yet how long it may take, what the recognition might look like, what's going to be in their new revision of the use of 1093-1 guidance document. We don't have answers on a lot of that yet. We know from behind the scenes discussions that there may be certainly some non-recognition or at least partial recognition of some portions until it gets vetted out a little bit further, but that's not a firm thing that's known yet. But it hopefully doesn't quite lead to needing to have separate regulatory paths for different regions of the world, although it is a possibility. Thor?
Yeah, and just to add, so we went through these high-level possible issues that we want to address. Obviously, there's huge amount of discussion that goes into each one of them, and the attendees, the registrants, put in a bunch of questions that we will be addressing first, and then hopefully get to some of the questions that you submit during this. But we'll go into a little bit more detail in some of these just based off the questions that were submitted, and you know, because they'll hit on each one of these a little bit more. But if you haven't been up to date on what all encompasses, just so you know, there has been webinars done by many people, and especially here at Nelson, we'll provide links if you wanted to go back and actually watch and see the details of what's changing for each one of these. We've done webinars on it in the past. Today, we'll probably mostly focus on the issues that you guys have had in industry in general, the questions we've gotten, and try to inform as best we can on the current state of knowledge. One of the things I am going to talk about before we get to your questions, though, and this goes into a lot of what we've heard, which is what does this mean for regulatory acceptance or the U.S. position? I just want to kind of let's see how things progressed, right? So there were many votes through the committee drafts and moves to discs and FDIScs and FDIS acceptance. And you know, as you can see at the beginning, there was two votes as far as no on the first committee draft. And then the one consistent thing after that has been US. Now, with the FDIS acceptance, we did get a little bit more push as kind of concern on this draft, but it still nonetheless did proceed to acceptance, which means this is the document that we're going to have to go to. But as you can see, US voted no every single time. And there's a lot of reasons why US has voted no. I think just overall is something that Nick started off with, which is, and me personally, just so you know, I have voted no every time. And the main reason why is I just don't think this draft is ready yet. I think there's a lot of areas that need to be, guidance needs to be better given. We talked about risk estimation. I think foreseeable misuse, end of life. I think there's a lot of concepts that I don't necessarily disagree with, but right now I don't feel like there's enough guidance given to really have a understanding what's expected from the point of view. And I think there should have been more work to develop this this standard before it was released. Although that's now where we're at, we're releasing it. And like, Chris said, we are right now working on subcommittees to write either TISs, technical specifications, or technical reports to try to fill in these gaps that might later be a part of the standard. But I felt like they should have just waited and worked on those before they released the standard, or could have gone back to committee and worked on this. So that's why we voted no. That's why I voted no, at least. And I think at this point, we'll go ahead and head to questions. Brandon, I don't know if you wanted to kind of send out the first questions that we got on screen, and we can kind of talk through it as a team.
Yeah, of course. So one of the first questions we got, again, thank you everybody for submitting questions prior as well. So more risk-based assessments, does that mean I can rely more on historical materials or processing chemicals, less on test results?
So I'll take this one, and then I'd love to hear from you, both of you guys, on this. I think there's two things to think about. I think there's the theory of this document, and then there's the practice of this document. I think in theory, going more to 14971, having more of a risk-based approach, I think most people could reasonably say that medical devices have historically posed a pretty low risk. We're using materials that are pretty common in medical devices for the most part, processes that are very common throughout the industry, even suppliers that are pretty common for many medical devices. And so in concept, I think that that's the case. I think that this standard should allow you to do more of a risk. In fact, I feel like that's the spirit behind this new change of document. I think in practice, though, I think here's the problem with that concept. I think 99% of the medical device companies that I work with, we work with here in Nelson Labs, are very good at choosing safe, no materials, choosing practices like cleaning processes and cleaning validations and different types of techniques and manufacturing processes that lead to very little risk in medical devices. But an FDA person would always tell you they're not worried about that 99%. They're worried about the 1% of the people that try to cut corners or try to do things differently. And it's hard unless you have really good defined materials to do that successfully with the regulatory body, especially the FDA. And so I think practically you can still do it. We've had success doing it, but I think you're going to have to be really good at your material evaluation and your process evaluation and the impact of those processes on the material to actually be successful going down that path. Gentlemen, what do you think?
Yeah, I'll just add a quick piece of that too, Thor. I put kind of my shoes on when I worked in orthopedics, and my thought process is always, and I hope we get to this question around misuse, but my thought process is always there is so much design risk that is captured from the engineering perspective and the production perspective of our devices, right? So with that being said, more validated production lines, more validated cleaning processes and disinfecting processes, many operations taking place in controlled environments, including clean rooms, et cetera. We should be able to leverage a lot of the understanding of our materials and our processes along the way. So I do agree conceptually, you hope to see that. I wonder if we have enough written in the standard for regulators to see through that and not point to anything black and white that may be missing.
Yeah, I think in the immediacy, when you're making these changes to a device, when you go from version 1 to version 2 of your own device, where you've got a lot of comparative data that you can draw from, where you can just line it up version 1, version 2, and the differences, and every detail that's gone into it. This sets up really well for doing a risk evaluation of that and being able to potentially get out of doing testing and using risk assessment instead. It's when you're pulling from information where there might be data gaps because the predicate or the comparator device may not be your own, that there could potentially be challenges. But I do believe that the standard itself is moving into an environment where we pull more on information that's been previously gathered to support the future, because everyone is building with the most positive intents.
And Chris, actually, that brings that right here, this biological equivalence. The whole concept of that is exactly what you talked about. And I think that's easier to do on these changes. Well, let's face it, especially since, post-COVID, it seems like changes with supply chains and things like that have been something that we see more of. And bioequivalence is something you might be easier to do with a change in a device than a brand new device. I totally agree. Brandon?
Awesome. Another question here. Any impacts to particulate testing of devices?
Nick, what do you think there?
Yeah, that's an interesting one. So I'd love to hear Chris's response on this too, with Chris's background. But for me, it's interesting because we see so much particulate generation in the clinical use of these devices. And I already see Chris shaking his head, yes, in agreement here. But so particulate for me had always been a really hard one to spend a lot of time and energy justifying, right? Just the natural kind of process of implementing the use by clinicians of our devices, especially our higher risk devices that require surgical intervention, generates so much particulate, right? And so, it's interesting to me to make sure that we're able to identify the risk of particulate generation from our devices, what we know will happen from the devices based on those materials. But that kind of does go back to our original question of knowing our materials, knowing what's biologically inert, what is not inert, what will the body do with these materials, after, interaction of the device or clinical use of the device, and trying to defend, what it means to generate particulates and what that process looks like with the body too. That's my perspective. I love that approach. Curious, Chris, kind of what you'd have to add to that too.
Yeah, when we're talking about just the manufacturing of the device, I don't think much changes. We still need to consider the risk of any residual or particulates that don't come off as part of the normal manufacturing cleaning process. But I've seen over the last couple years, it's really stepped up on when we do need to do testing of the device. The historical way, there's in many cases we would do a cut and cover. So we would cut the device into little pieces and then proceed with testing. And that process in and of itself, it's rather easy to produce a fragment of the device that it's hard to differentiate. Is it a particulate that's from the extraction process or some residual from the device, or is it just a part that was from cutting? But then further is the extraction environments themselves are intended to mimic, but clinical environments, but not always. And so it's really seen a big uptick on the necessity that when particulates are seen in some of these extracts, that we do need to identify what are they, where did they come from, could this happen in a clinical environment or not? Because again, the device may not be exposed to sodium chloride clinically, and that makes 304 stainless rust. And so if it can or can't happen clinically or not, if it could, well, what does it mean to device performance and safety? And so there's been a real big emphasis of the last few years anyway to further push on proof of some of these pieces, not just, I think it's from X, Y, or Z. And more companies, I think, are moving to an environment where they're not cutting their devices. They're testing them intact, in particular to not expose surfaces that shouldn't be, but to prevent from some of these, let's call them artificial, non-test article induced changes.
Yeah, and with that too, it also, just to add, it kind of raises always the question of, are we generating more particulate just through the sample prep process, prior to testing than we are in any clinical environment per our device's intended use? So there's always that consideration, and always a really critical factor in my opinion, in vetting our testing laboratories and testing partners and making sure that you have a very good sample prep plan for these specific devices where that could be a bigger risk.
I think there's two things to think about here. One is definitely the particulate generation in the sample prep that would then make a regulatory body go, well, why is your device shedding particulates? And then I think there's what's the new section in the new 10993, which is, I think for the first time ever, there's some real direction on when to consider particulates, right? So if you have a cardiovascular device, I think that now we have to start to think about the risk of having particulates being shed into the blood system. Or if you have an orthopedic, what's the risk of having shedding of particulates in a joint, right? And I think that goes to risk assessments, but I also think that is something that we don't traditionally, we have done for medical devices, but we don't really have an acceptance criteria, right? So I think most labs use the USP acceptance criteria for injectables, which doesn't necessarily mimic what would happen with a medical device or the concerns of a medical device. So I think, once again, having more kind of direction on what to worry about, but not really giving kind of like the practicality and how to do it is something that I'm a little bit worried about going forward too.
Okay, we got another question here. Will FDA or Japan accept not performing rabbit pyrogenicity tests?
Pyrogenicity.
Yeah, so I'll take this one and I'd like to hear you guys' thoughts. So, okay, so the FDA is the one that helped write this section. And when I say eliminate rabbit pyrogen test, it's not really eliminate the material. We didn't remove it, right? The verbiage was compromised with the FDA, which was that as long as it's not a novel material, you can risk assess your material-mediated pyrogen through written justification. So the FDA is the one that contributed with that verbiage. Now, like Chris said, we're still waiting to see what parts the FDA will recognize and not recognize. But I can tell you the FDA will help work on this section, and with some help from the industry and certain individuals on the ISO committee by submitting data from E&L studies and different types of rabbit pyrogen studies. I think it helped convince a lot of the scientists there that material-mediated pyrogenicity really isn't much of a concern for medical devices. Now, Asia, they were part of the discussion. They voted yes, but like Japan, China, it's hard to know exactly what they will accept and what they will not until we start getting submissions there. But I can tell you that all three of those countries were part of the discussions and they all voted on this document. But the FDA also was integral in contributing to the language of this section.
And what's in there does align with, in our ISO working group 16 on pyrogenicity, that is the general sort of consensus is in alignment with the article that was recently published also in Alt Talks about the pyrogens not being as readily present in medical devices.
Yeah, Kelly Copeman and Lisa's article is the one that most of this was built upon. So,
Yep, agreed with it all.
Okay, Brandon.
Okay, another one here. How does the requirement for author qualifications relate to performing gap analysis on BP, BR, BRA, and TRAs?
Chris, you want to lead off this one?
Yeah. So the gap analysis generally falls into two buckets. We need to understand in the standard itself what the changes were, and we talk about a lot of these things and what we're talking about today in the prior webinar that got linked in the chat. But so we need to understand from the standard what changed, but then we also need to look back with how does it affect those previously written documents, and then even potentially some of the test reports that those were built upon, to make sure that that test report is done to current standard and that the conclusions that were based upon those test reports, the risk assessments, the material evaluations, the extractables data, and so on, then further step up into where we are today. So it's a multi-step process. And in some cases with the qualifications is that, yes, you could have a good biological assessor be able to do all of that, or they might have a team of individuals who would contribute to that. But the risk assessment, I think, does sort of cover those threefold steps, from the evaluations that were done to the BEP, the BER, and then making sure that they conform to today.
The only thing I would add is what Chris talked about when he got here. This is definitely more of a concern from notified bodies than from FDA. they're the ones that pushed hard to get this beefed up. So I think it has to deal more with when you submit to your notified body on the person who's writing the document.
Yeah, and I just want to add to that too, just keep this brief, but I think we all probably saw this coming, right? There was a really big push on risk assessments in the recent years, specifically from the FDA, on a true written plan, right? Now we're talking reverse 7 to 10 years ago when we were, really priding ourselves as industry saying we're moving away from checking boxes, right? From perhaps quality or regulatory or even engineer officials within an organization downloading the standard document, looking at Annex 1, checking the box for the test report, submitting to the lab, and then adding that to the 510K, right? We've come a long way from that in, I want to say just 10 years, but that has been a long time. So we've come a long way from that, but now I think it is time for us to roll back our understanding of what is a good risk assessment. And I think that's what we're getting to here at the end of the day. I'm not saying, hey, we're going to require you now to outsource this process or to hire more qualified individuals. That may be part of it. 1 or 2 of those may be part of this moving forward for particular organizations. But at the core of this change is resulting to more appropriate risk assessments that have better scientific knowledge, better understanding of these processes, especially as we move to where we hope to be, less invasive, less testing in animals, more in vitro based, leveraging chemistry and potentially in silico models to come. We want to make sure the right people are really signing off and understanding that and the potential impacts and risk mitigation for these devices. So my addition there is just, I think it's deeper than just hiring the qualification standards.
Good point, Nick.
Okay, to look a little deeper on those BEPs and BERs, do you anticipate more pushback from EU regulatory bodies on BEPs and BERs, which default to testing in lieu of risk-based approaches?
Good question. So I think this new standard makes it more difficult to write a BEP, right? So let me tell you why. We have to worry about foreseeable misuse now. We have to worry about life cycle evaluation, which I hope we get to those questions when we get there, right? So we have to look at risk estimation as part of a BEP today, according to this new standard. And that's, and to Nick's point, that's getting way away from just checkboxing tests. So I don't know if they necessarily are going to just automatically be more like thoughtful on their evaluation. I think the new standard requires more evaluation and thoughtfulness just based on the requirements. And so like someone has to really think about foreseeable misuse and have to be able to defend what they think is foreseeable misuse to the regulatory body. So I just think that's inherently more of a justification discussion kind of BEP than traditional. You guys have anything you want to add? Or we're good?
Yeah, I agree. And especially in the environments where you do need to bring in multiple experts from multiple different groups, it's really going to be a team approach from really getting those material evaluations and digging through suppliers, working with the regulatory folks on your pathways, working with the whoever the biological assessor is who does have the experience and digging and asking the right questions to the right folks and the clinicians and so on.
Yeah, I'm good with that.
Great. For another one here, what is your intake for bioaccumulative chemicals? The draft states device contains bioaccumulative chemicals should be considered long-term contact. Will this change the test requirements? For example, do I need implant study for a limited contact catheter?
Chris and Nick, I'll let you guys start on this one. I have my thoughts, but
yeah. So I think the big thing comes for when you have a repeat use device. So the first time that was really put down on paper that I'm aware of was in FDA's pulse oximeter guidance, where they talk about how we start to think about repeat use products, where a single exposure is a full day, no matter how long, and that time in between uses might need to be considered continual contact, even though it's taken off and put back on. But then also for these absorbable, degradable materials, that they now have the potential to bioaccumulate in the body somewhere. They're not just, once it's absorbed, a wound gel is absorbed, it's not magically gone and metabolized instantly. It persists within the body longer. And so for a solid device like a catheter, I think it's more a consideration on if there are coatings that might come off that have the potential to bioaccumulate in the vasculature, in the kidneys, and the liver, or so on, and need to work on it from that perspective. Solid materials, I think, will be a little bit easier if they're just a limited single use. But as soon as you get into the more repeat use, or there's contact agents that might come off, is where things will really change, and the scope of and they're not endpoints anymore, they're biological effects. Those individual assessments that we look at, irritation, sensitization, hemocompatibility, that's really where we need to consider those, is when things will persist in the body longer or have a longer duration than previously we had considered.
Yeah, I think practically, I think Chris is right. So there are going to be some materials that are concerns. PFAS is starting to become something everybody's thinking about, right? So you start looking at, do I have anything on my device that I know will bioaccumulate when it gets in? And there could be some evaluations that you might have to do with that. But I think and then if it's a absorbable device or a solution or gel, those things might be a little bit different. I think for the majority of devices out there, this really goes into why that's the recategorization of the repeat use. And so for those of you that may not be aware, right? So a lot of companies historically, if your device had contacted the patient for an hour a day, but for 5 days in a row, a lot of companies would just say, okay, it's an hour times 5, it's 5-hour contact, it's limited contact. Well, like Chris said, now in this standard, regardless of contact duration, it's considered a day contact. So that same device now would be 5 days, which is prolonged. So, and the reason why the justification of that is this, bioaccumulation concept. So almost you'd have to justify that you do not bioaccumulate more than 24 hours in all your materials and compounds to be able to even go back to categorize as limited, which is just a huge lift, right? So most pragmatically, most people are just going to start to do this new type of categorization to deal with bioaccumulation, unless you know something's in there that could have long-term bioaccumulation effects.
Yeah, my only addition there is just, I think Thor mentioned, the most critical part of that was justifying that the material does not bioaccumulate within the 24-hour window, right? So I think that it could be a heavy lift for some materials, right, to understand that. So again, this is kind of one of those in theory versus what's practical, right? What will notified bodies and the regulatory agencies accept for this in its place, or to support, meeting this requirement? And I think we'll soon start to see what that looks like.
And there was discussions in this, just so you know, there was discussions about, should we start looking at more PK studies, physicokinetic studies? That would be tough in my opinion, just because we don't typically have ever thought about doing those for med devices. But in some cases, with some types of these materials that we are unsure about, we might want to look at some kind of physical kinetic study. Where does it go? Where does it accumulate? How quickly does it excrete from the body? Those concepts, we might start looking at more and more of those depending on the materials.
Yeah, and the drug and chemical world is way ahead on this. They've accumulated an awful lot of data on these as out of requirement, but not all the materials and compounds that are in medical devices have been evaluated in this manner before, so we don't necessarily have data on all these compounds. Some we will, but even if we do, is it on the same exposure route, dermal in a wound versus systemically IV, for instance?
Yeah, or
orally for that matter, right? And so we don't is unfortunately an awful lot of unknown, and it's going to take some good toxicological thought, and we may end up with having to do some metabolism work potentially on some of these higher risk materials that have more unknowns with them.
Awesome. We've got lots of questions here, so we'll see how many we can get to. But the next question here is, can you guys explain how genotoxicity is going to be evaluated moving forward or being revised? There are several tests for this endpoint. Is there a good understanding which ones should be considered more than others?
Yeah, so Nelson Labs has this great slide ruler that we've been doing for years. We're having an update one coming up. But if you have one of those, you can pull it out and play with me. But basically, if you see and evaluate on anything that says systemic toxicity on it, then you have to consider genotoxicity. Now, most of the time, that's now looking at like short-term contacting circulating blood devices, but there's a few others. But the concept there is that if we're worried about a systemic exposure, then we should also be worried about genotoxicity. I have my feelings about that personally. I don't love adding test categories without like real data to help back it up, but that's how this came about. There was, the rationalization is that if you're worried about systemic exposure, then you should also be worried about genotoxicity exposure. You can do that two ways. You can do that through in vitro testing. So you have like an Ames test, a mouse chromosome aberration test, it's a cell test, a mouse lymphoma test, which is also a cell test, mouse micronucleus, which both has an in vitro and in vivo option, or you can do it through extractables and leachables. My recommendation is probably based off of both the scientific concepts, but also your business concepts. Which one to choose kind of depends on what other endpoints you need to evaluate. Does it make more sense from a cost and timeline to do the in vitro tests, or are you already doing the extractables and leachables, and can you cover that with your ENL? But that's kind of an overview of why it's been added and what devices are going to be impacted. Nick and Chris, you got something you want to add there?
Yeah, for me, it's just you nailed the head, hit the nail on the head there, Thor. If you're opening up to systemic toxicity, you need to understand what genotoxicity risks need to be mitigated. That said, for the last few years, we've been really focused as an industry on conducting the appropriate extractable leachable chemistry to be able to mitigate those risks of genotoxicity. So now that doesn't mean that any ENL chemistry can mitigate and serve the purpose of confirming no adverse effects from a genotoxic perspective, but the appropriately designed study certainly can. And so, we often recommend this at Nelson Labs, but if you have a very novel material, novel device, really interesting, complex device, the appropriate ENL study and approach to be discussed in a pre-sub meeting with the FDA goes a long way. Right, so that is where it's worth spending the time and energy from your regulatory and quality group within the organization to make that effort to try to design those studies as early as possible. Again, for more complex devices, but otherwise, yeah, I agree with what you said.
And just to add before Chris goes, I also want to say it's like I don't want you to automatically think about testing either. The concept here is evaluate genotoxicity. So can we evaluate that through other means besides testing? It's possible. We've done it here at Nelson before. So don't always jump to testing, although if you're going to do testing, that's the decision between in vitro testing or ENL. Sorry, Chris.
Nope, nothing more.
Okay, thanks. Brandon?
To elaborate a little further on genotoxicity, it was considered long-term exposure risk for so long, but now it's considered a short-term risk. Is there new data behind that?
So kind of talking to what my first comment on this, just so you guys know, was that it takes an act of God for us to remove a test from 10993. Look at material-mediated pyrogenicity. We have to have so much data, so much work to be done. Even to try to change a test from in vivo to in vitro, it takes so much data. but yet we can add one just by saying that we should have a concern. And I felt like that was kind of backwards, that we should probably have some kind of data to help support that there is an industry concern. To be fair, genotoxicity is a very hard endpoint to get data for. What clinical result would express itself through genotoxicity, at least quick enough and close enough to the patient's the device to be able to attribute that to the device. So there could be clinical issues for genotoxicity, but we just might not even know it, right? So to be fair, that's hard to do, but I did put that comment in. So Chris and Nick, I don't know what you guys think.
I agree. It's hard to necessarily attribute causation of genotoxic cancer type effects to the device unless you can do very specific clinical testing on that. But I think it was more of a general concern and a little quick to add, but we do have risk assessment ways to get out of it.
Yeah, I just think this is one of these areas of the standard that probably should have taken more time to understand its full effects, because, to Chris's point, is there a way to understand those effects of particular, compounds and uses? Yes, but those studies are not what we're used to doing in the medical device industry. And so again, it's kind of bringing in this whole perspective of, well, I feel like I need to test to meet these requirements. That's not always the case, right? And let's also remember part 2, where we do want to focus on animal welfare as well in instances where we can justify elsewhere. So it does create a little bit of what I would consider here unnecessary complexity to risk assessments around this area.
All right, great. Going back to some of that foreseeable misuse, can you guys elaborate on the foreseeable misuse and where do you draw the line?
I'll let you guys go on this one first. I knew this was going to be a big one.
Go ahead, Nick.
Yeah, so interesting. I touched on this a little bit before. My background in training kind of tells me that a lot of this should be determined in the design risk assessment of a device as well. More of that has been focused on failure modes. And so I think we see a lot of risk categorization and mitigation in device failure, but we have historically had an issue with that coming from understanding what potentially misuse can be added there as well. And so for me, misuse is really understanding what could happen with the devices, right? Are these devices going to be used off-label in different applications that we know are prone to use depending on how they were designed and how they're distributed, right? In what particular industry they're used and why. So I think understanding that is kind of part 1. Part 2 is also making sure that we're really, really clear on our device indications on our DFU and IFUs. And I think this opens up a little bit more interpretation to leverage those DFUs and IFUs to show that this is what the device is indicated for. And because of these risks, elsewhere, we have not kind of explored any other options for, what potential misuse risks do align with the device. So I think right now, for me, the way I see this, until I see more feedback kind of gathered, is, misuse really is in line with what we already understand the devices to be used as outside of their intended use, if you will.
Yeah. And so I think there's 2 other pieces to add here. And while it's labeled as foreseeable misuse, I think it also can be categorized as being exposed to other clinical environments that we hadn't previously considered.
Yes.
And so I think of misuse being a intentional thing, but it could just be that, hey, I thought my device was ordinarily in almost every case going to be used on intact skin. But I suppose that it could be plausible that someone could have a trauma to their hand and that the device would have to be used to extricate something from their hand which had an open wound present. And so it's not really a misuse, but it's an alternative clinical environment that we hadn't previously considered. The other piece is that I came across a number of folks who, when they look at the scope of the evaluations they have to do, if they were to categorize their device as prolonged versus long-term contact, it opens up the length and duration. If they need to do testing, the length and duration of what they have to do can extend for months and months and months longer. And their regulatory pathway can become more challenging. And so what they said they would do is they would put labeling on the device to only use it for up to 30 days, just to get through their submission easier, knowing full well that clinically it was going to be used for months and months and months longer. So it was sort of skirting around the rules a little bit. So I think part of this is also trying to wrangle industry a little bit to consider the actual use and not some of those, what we'll call misuses in that case, where you're labeling around a known scenario.
I 100% agree with you, Chris. In fact, that's why this was put in here. We do this as human beings all the time. People who wear contacts, they said, okay, you do not sleep in this contact or whatever, but you know, people do, or they wear it. It's like we push the boundaries. How many times, you know, you open up your yogurt, it's past expiration date, but you know, it smells okay, you're gonna eat it anyway. We do this. And to Chris's point, in my career, I've seen tons of people either, you know, contraindicate to a population that they don't want to evaluate, or length of time they don't want to evaluate, but they know that doctors are going to do it that way. And this was put in to try to kind of go against that concept. Like if you know it's going to be misused in either contact duration or location in the body, you have to evaluate it. The thing I'm scared about, though, and by the way, this is another one of those areas where we have a working group that's writing for another document, is we don't really spell out exactly. We talk a little bit in there. If you read the FDIS, when it's soon to be standard, it talks about those as far as length of time and contact duration, but doesn't really give a lot of other guidance. And so, man, I'm scared of what my regulatory bodies are going to think about my product being misused. So my biggest fear is if you're doing extractable leachables, you set your AET based off of a TTC, which is based off of exposure time, and then also some populations. And so you set your AET, you do your chemistry, you evaluate their compounds above that AET or TCL or TLC or whatever, TCL, and then you submit that to your regulatory body. They come back and they say, wait, I think this could be used on a different population or a different length of time. Now we're going to have to go back and reevaluate. We have to drop that AET. And then it takes time. Like, that's what scares me about this. I hope we can get to a good point sometime to kind of understand what this is going to do. But I think this could be an open-ended issue just based on your regulatory body.
Yeah, this just certainly seems like, I hate using this terminology, but the wild, wild west, you know, here, kind of opening up to a lot of interpretation of what misuse looks like too. And I think this would be very dependent on the device type and the categorization. I think that's where we're going to see a lot of potential different feedback is, again, where the device is used and what it is.
And before Brandon jumps in, I think we're going to be getting out of time. What I would like to do too is, we have all these questions that have been submitted. I mean, tons of questions. Chris and Nick and I will go through it and we'll try to answer them in some kind of document that we'll put onto the Academy and on the websites, just to try to get some frequently asked questions out there to you guys, because there's no way we're going to be able to answer. I think we probably have time for one more, Brandon, or something, but I want to make sure we try to answer as much as we can.
Yeah, we do have time for about one more question. I think this is a great question to wrap it up to. Is there a time frame for medical device companies to convert to this new ISO, and how would one go about performing a gap assessment for this update, because the standard is a complete rewrite?
Chris, you want to start this one off?
Yeah, I think we touched on it a little bit, but expect that there are regions like Europe who will expect you to conform straight off the get-go. Now, if it's not a published standard yet, how are you going to know the full content, I guess, unless you listen to talks like this, which help inform, or if you're on a standards committee, you'll have a little bit of a heads up. But expect that they will want you to conform almost immediately. And Thor, I think you said that you talked to a couple notified bodies who said, yes, we want you jumping in head first straight off the get.
State of the art.
The other piece, go ahead.
They said state of the art.
Yep, SOTA, state of the art. So we need to also consider that from the FDA, that may take a while so we understand where things will be at. And so typically also with FDA is that whenever there's a new standard, no matter what it is, they have their recognition process, or their FDA recognized consensus standards, in which they'll include the parts for anything that they agree with or they don't agree with, and they're exempting their recognition to. But they also is a grace period in there where they say you can follow the old standard up into a certain point. Now, we know from practice that there are reviewers who, sort of like Europe, want you to start conforming sooner than later, but the theory is that you do have a little bit of a grace period there. Now, if you're going to go into multiple regions, that's where it can get a little bit tricky, knowing that Europe, for instance, may want the whole kit and caboodle, but the US FDA may only require a subset. That's going to be a little bit harder, and we may need to go back and do some updates to some BEPs or some BERs or so on, and do some amendments to them to help you conform. But there's an avenue that we can to go back. But if you're going to Europe first, you're sort of going head first all the way in, and then if FDA recognizes only a subset, then we can step back from that a little bit and do some amendments to hopefully address those.
And I expect the FDA to partially recognize this document. I don't know that for sure, but that's what my feeling is. And most of my contacts there have not told me one way or another that. It's just kind of working through this process for the last couple of years. I do not expect full recognition for sure. I do think there'll be some partial, but to Chris's point, I think they'll release their own guidance document on how to interpret. I think there's going to be things in this document that they will definitely have in there, but that's going to be something we're going to all have to see together as far as what the FDA's point of view. But yeah, you saw 7 to 8 weeks is what we had from release. A guinea pig maximization is about 8 to 9 weeks. So you can understand that you got that much time to basically start the testing and move forward. And so if you're going to Europe, you might want to start thinking about some of these things now.
Yeah, the only thing I would add to that is I think that is probably the silver bullet question of the afternoon here too is, when should I start, paying attention to this and implementing it in my projects? The answer is now. If you're working on a project right now, if, a design project's hitting your desk, if you're working on a risk assessment for a device, I would certainly ask your testing partners, your toxicologists, your biological assessors, to consider the changes in this document moving forward.
Great. I'd like to thank you guys. We are out of time. I think that was a great question to wrap up on. And again, thank you, Thor, Chris, and Nicholas, for your insights. And again, thank you for everybody for joining us today. We will be putting a frequently asked questions out, like Thor mentioned earlier, on the Sotera Academy. So keep an eye out on your emails for that. So thank you again, and everybody have a great day.
All right. Thank you, everyone.
Thanks, guys.
Thanks, everyone.